Present on our green dSTORM technology, and this is thanks to a new buffer that we developed that we called Eternity Plus. So to start with, I want to illustrate the super resolution approaches that we actually have available in Lyon. And for that, I want to take for the reference structure that we will be looking at is actually the centrosome, which is an organelle in the cytoplasm of our cells. And we will be looking specifically at the distal marker, which is highlighted here in purple. And so if we want to look at this distal marker by confocal, this is what we get. You have the scale here, 200 nanometer. And then if we look at this guy by 3D SIM, which is one of the super resolution approaches, the structured illumination microscopy, 3D SIM, this is what we get. So we start to get some structure with a donut. And the advantage is that we can increase the resolution by a factor of 2 and be able to look at quite a lot of colors still. Then there is the 3D STED that we have also available in Lyon. We have an Abereo system. So here you can see actually a resolution of about 3 times, depending if you are in 2D STED or in 3D STED. And you can still look at about 3 colors. And then we are mostly interested by single molecule localization approaches with dSTORM. And this is where you get actually for those techniques 10 times to 20 times resolution compared to the confocal. But there is a limitation in the number of targets you will be able to detect. On most commercial systems, it's 2 colors. And so for that, we have 2 systems in Lyon, one from Zeiss and one from Nikon, and actually also one right now from Bruker as a demo system. So those approaches can be actually increased even more when we use mean flux approaches, which is another approach. I'm just mentioning that because we also did images with this CEP164 distal marker. And as a reference, I put here the electron microscopy image you can get by TEM. This is from a previous paper from 1992, actually from Vintran. And then there is also an interesting way to use is to do actually expansion. So these are the 3 images here, where you can actually recognize now pretty easily the 9 structure, the 9 distal appendages that you can see already in the electron microscope picture. And you can see them already in white field here, confocal mode and 3D STED mode coupled to expansion. And you can actually see that those 2 guys, expansion STED and influx, start to look quite similar. For sure, it's not correlative. It's not the same centrosome and in the same cells. And actually, those expansion images have been made recently in collaboration with Karsten Jenke and Maria Genova at Curie Institute. Okay, so what I want to present today is results that we collected through a CNRS prematuration project on super resolution dSTORM in 3D and in 3 color applied to centrosome orientation. And for that, so I'm working with 2 other colleagues, Christophe Plas, also in Lyon in a physics lab, and Arnaud Fabier in a chemistry lab. And we also hired Corentin Rousset as an engineer to work on these projects for 2 years. I will present you this project. So first, to start with some biology. So we are interested by a protein that is called nucleolin. That is a multi-dimensional protein, and that was first described to be localized in the nucleolus, and it's involved in many processes associated to nuclear function, but also transcription, translation. And we found actually that it's also associated with the centrosome and specifically the mature centriole in our cells. also a role, potential role on microtubule dynamics, and a little focus also on plasma membrane, where it's able to actually target some agents like some viruses in the cell. Okay, I don't want to do a full review on nuclear link because there is a lot of stuff on this multi-dimensional protein, but focus on the centrosome. We actually published 3 papers where we found that silencing of nucleolin leads to amplification of immature centrioles, that it's also modulating microtubule anchoring at centrosome, and modulation of microtubule nucleation speed. And just to do a little focus on centrosome ultrastructure, this is so 2 centrioles seen by electron microscopy. So there is one that is here and one that is here, and you will understand a little bit better how it's organized and the size of those guys. So you can see that here it's about 450 nanometer, and in this direction 250 nanometers. So that's why also we need these super resolution techniques to look at the centrosomes. And what I can tell you is that at the basis here, you have what is called the PCM, so pericentriolar matrix, where the microtubule are actually nucleating. And then they are anchored at the subdistal appendages that I highlighted here in red. And the subdistal in red are for microtubule anchoring, and the distal in orange are important and essential for cilium growth during differentiation. Okay, and so you can see the difference between those 2 centrioles. So this one is the mature one because it has the distal and subdistal appendages, and this one doesn't have yet the appendages, so it's the immature centriole. Okay, so we started to do some localization of different components of the centrosome. So here in purple, here you see a CEP164 corresponding to the distal appendages, and in orange and seen in blue here, the CEP170 appendages that are just below the CEP164 that are important for microtubule nucleation, sorry. So this is one color for each of them in dSTORM. The scale bar is here, 200 nanometer. And you can also do for sure 2 colors, looking at one crown of appendages with another component of those centrosomes. So it's interesting to see how they are localized according to one another. And for sure, as you know, it will help us to also understand their function. Okay, so what is the principle of dSTORM? The idea with this movie realized with the Eiffel Tower is to actually have our fluorescence that is actually blinking. I will play again the movie after. Then you can actually detect the different molecules that are blinking, and you will reconstruct a super resolution image. I will play it again for you to show. So, and you can see that then there is more and more spots here to reconstruct your final image that is better resolved than it was for sure when it was just blinking like that. And the idea is to actually detect the gravity center and to project them on one single image that actually is referred as the super resolution image. So this is the principle of direct stochastic optical reconstruction microscopy. To reach, sorry, I want to go out of that and go to the next step. There was one more thing I wanted to show you, which is here. So to get to this blinking phenomenon, you need dSTORM buffer that is composed of a reducer, which can be twofold, MEA or BME. You need to be in absence of oxygen. This is the way to get into this dark state and then reach an equilibrium where you see this blinking. And you also need high laser intensity. Okay, this was the development of dSTORM. It was with this buffer. There is one limitation to that buffer, is that there is a very short lifespan for this buffer when the lack of oxygen is obtained with enzymatic system. Okay, it's only few hours that you have to look at your samples, and when it's done, you have to unmount your sites and mount them again with another fresh buffer. So we decided to actually work on that limitation, and we actually found that if you remove your oxygen physically, then you get a buffer that we called Eternity. That is our first innovation before starting this project, and you can observe your samples during several weeks. And I want to illustrate that again with the centrosome labeling. So we like a lot of people, the reference fluorochrome is Alexa Fluor 647 to obtain this good blinking.